Hey guys, welcome back to MicroJai, where we do all things microbiology, and today we're going to look at yeast cells. So I had two different kinds of yeast. I had the store brand, like the 99-cent one, and then I had the $5 one. So I put the yeast in warm water and I added a little bit of sugar, and I added a little bit of sugar, and these are the results. I let them sit in the warm sugar water for about 45 minutes. So this is the store-bought one. So quite a bit of bubbles. So this one is the name brand one. It definitely overflowed twice. So if you're baking bread or anything, I definitely suggest the name brand one. So to prepare my slide, I'm going to take a microscope slide, fresh plastic pipettes here, and I'm going to try to get the water, because all of this are bubbles, so I'm gonna try to get the water at the bottom. Only need a drop or two. Let's do 2 drops. All this is trash. And then I'm gonna take a cover slip. I'm gonna take a cover slip and put it right on top, and then put it on the microscope. There's a hair here. Let's remove that. Clip it on, clip it on the microscope side, and attach my phone. I'm gonna start with the 4X. Find good spots. So these are the yeast cells at 4X. I'm gonna go to 10. Okay, this is how they look. They just look like circles. I'm gonna go to 40X. My 40X only works with immersion oil for some reason. I don't know why. But usually you use immersion oil for 100X, but I've learned that my 40X likes immersion oil also. So I'm gonna go to 40 for a total of 400X. Okay, here are the yeast cells. They are kind of ovalish shape. You see them moving. Let's go around. Yeast are unicellular fungi. So they're a type of fungus, and their scientific name is Saccharomyces cerevisiae. And this species of yeast is used in winemaking, brewery, and baking. So these yeast cells double every 1.5 hours, roughly 90 minutes. And believe it or not, yeast are the first eukaryotes whose genome was completely sequenced. So yeast were kind of the pioneers of genomics. So yeast can be found in warm, moist environments. And let's get a little closer here. Okay. So these are the yeast cells close up. Oop. So the one in the middle. So yeast cells reproduce asexually, meaning by themselves pretty much. And how they reproduce is through a process called budding. That's the type of asexual reproduction that they do. So what they do is kind of like form a whole nother cell with this. So this tiny cell attached to it, that's another cell that they're forming into. So I was able to capture the budding process amongst yeast cells. So these two cells are finishing the budding reproduction. So the cell on the left, it just completed cytokinesis. That's the separation of the cytoplasm. So the baby cell is all free and moving from the mom, going to do what yeast do, and go produce carbon dioxide or something somewhere. So how yeast cells work. So yeast feed on sugars and starches. So when you're baking, you put yeast in warm water and sugar. So what they do is they feed on the sugars and starches, and then they convert that into energy, and then they produce carbon dioxide as a byproduct. So basically they eat sugars and starches and they fart out carbon dioxide pretty much. So I was trying to record those two cells complete the budding process, but then I saw like that cell right there moving pretty fast, and I see all these other cells, and I was like, but what happened was that thing right there, that is actually a carbon dioxide bubble that they produce, and it's pushing all of the cells towards the sides of the slide. Yeast genome consists of approximately 6,000 genes. As compared to a human, we have roughly 20,000 genes. They play a huge part in biotechnology. So because their genes are completely sequenced, scientists can figure out how to maneuver, how to switch some genes around in order to do stuff. But thanks for watching. I hope you learned a bit about yeast cells and what they do and how they contribute to brewery, winery, and bakery. Please let me know what to do next, and thanks for watching. Please like and subscribe and comment, and have a good one.